Good afternoon. We will continue our discussion on biotechnology. So at the end of last class, we were talking about the restriction enzyme. Restriction enzymes are enzymes that are going to recognize particular DNA sequences that are palindromes. So they read the same forward and backwards, and they will cut it. Either they cut it straight down in the middle and create blunt ends, or they give a zigzag cut and create what we call sticky ends. Right? So we were also talking about how to mass produce a particular protein, right? So first we look up the location of the gene using the online browser, genome browser it's called, and then we're gonna do PCR to clone it, and then now we have to insert that gene into a plasmid. So let's take a look at that today. Okay, inserting a gene into a plasmid. Okay. So here is a plasmid. I'll tell you more about the plasmid later on, but here's the plasmid. It's just circular DNA, and you can buy a plasmid online. There are like biochemical company, biotech company that sells them. Okay, so that is a plasmid right there. And just to keep it simple, let's say there is a palindromic sequence here. AAA TTT. Okay. And on the other end, it would be TTT AAA. Let's say this is the recognition site for restriction enzyme Z. Okay, I just make this up. And so we'll call like RE restriction enzyme Z. So what we can do is we are gonna take this plasmid and you know, if you need to visualize this, it comes in a little tube like that, okay? It's called an Eppendorf tube, and there is a little cap at the end. So you open up the cap, and then here you would have your plasma in a solution, right? The Eppendorf tube is really small. It's like this, okay? So you use a special pipette. You can take some enzymes, and then you're gonna put it in. So you're gonna add the restriction enzyme Z, and what happens is the plasmid is basically gonna pop open. Okay, let me see if I can draw this a little bit nicer. Give me a second here. It's gonna pop open, and we're gonna have oops, wrong color. We're gonna have AA and then TTTA like that. And on the other side, we're gonna have TTTA and then AA. Now it's a little bit difficult to see, so I'm gonna put a box around it. So do you see that these are sticky ends? They are sticky because they can actually reconnect. Take a look here. The A over here and the T, it's complementary to the T and the A. So the plasma can actually close and reconnect. Is that okay so far? So you take the plasmid with a particular restriction enzyme site. You're gonna use the restriction enzyme to cut it, and now the plasmid pops open.
Professor, excuse me, what about C and G? It should be 3T, 3A?
Oh, I mean, it could be anything, right? It could be like there could be C and G as well, but like
Oh,
to keep it simple, I just make this up.
oh, it's
Yeah,
an
yeah,
example.
yeah.
Okay,
This
thank
is
you.
a palindrome, right?
Okay.
All right. So now I have this cut plasmid in the tube, in the Eppendorf tube. Okay. Let's mm, leave that alone for now. We're gonna come over here and we will have another Eppendorf tube. This is straight out from the PCR machine. Okay? So this is the gene of interest. Cloned by PCR. Maybe I show you what a tube is so you know what I'm talking about. Hold on, just keep drawing. Here it is, okay? It's called an Eppendorf tube, and it's like that. They're very small, okay? This small. And you can do a bunch of stuff with them. Okay, so in this tube, you're gonna have the gene of interest. Okay, so that's gonna be the gene. Now, yes, we copy the gene, but then the gene also has the promoter and other stuff, right? So there might be some additional DNA on either side. And if the we call them the flanking sequence, okay? If the flanking sequence also have the same recognition site, they also have AAA, TTT, like this. So this here is the gene that you're interested in. And then let's say, right, the flanking sequence contains restriction site. 4 has to be the same enzyme. Right? The enzyme is restriction enzyme Z, right? And you will see why this is important in a bit. Right, so what we're gonna do is, remember they're all in the tube, and you can add the restriction enzyme Z, then you will, oops. So this is the gene again. But now, if you add the restriction enzyme Z, it's gonna cut it, right? It's gonna cut it just like over there. So there's gonna be a cut right here, and then we're gonna have a cut again. All right. So what happens is you will have the T T T A, and then AA. And on the other side, you will have the AA, and then T T T A. All right. So I'll draw it out for you so it looks more clear. Okay, so again, this is a sticky end. I'm gonna pause for a moment, let you catch up, and ask me anything. Anybody have any question?
Professor, these are all in chromosome?
These are all in well, this is only the gene that we copy, right? Because
Yeah,
we
before
wanted
we
to make the insulin, right? So that this is the insulin gene.
Okay.
Mhm. Any other question?
Yes, Professor.
Yeah, go ahead.
So, sticky enders, why they open and close? They exchange something or
It's not they exchange something. It's the sticky ends are complementary to each other, right? Like
Mhm.
this is TA and this is AT, so they're
Yes.
complementary, and that's why they can stick back together. Think of them as like Velcro. You know Velcros, like the one on the shoes that you can
Yes.
peel. Yeah, so you can peel them off and then you can stick it back on, right?
Okay.
Because they're complementary to each other.
Thanks.
All right. Okay, so what we do next is we actually take the content of this tube and then we mix it with the content of that tube. Right? So we mix them together. Mix. And in the end, you will get this. And then you'll get this. See if you understand it. There is gonna be the plasmid. I'm gonna erase a little bit of it. Okay. And basically, okay, please watch. Basically, this part is going to stick to that, right? And then this part is gonna stick to that. And then the gene is gonna be inserted. This is the gene. And then now you would have your sequences back together. Okay. So the sticky end of the flanking sequence of the stuff on the side of the gene, will attach to the sticky ends in the plasmid. The gene is inserted into the plasmid. That's all. Okay. But the gene might pop out again unless we glue it together. So what enzyme do we use to glue things together? Anybody? You know this. We learned this before.
Polymerase 3.
Polymerase will build DNA. We're not trying to build DNA. We're just trying to glue them together, join the fragments together. What enzyme does that?
Gyrase?
The ligase, that's right. The ligase. Okay. So we're going to add ligase.
So, Miss Vincent, I have a question. We took just one gene, or it happens many times? So this circle shows one of the cells in plasma,
Sorry,
or
one more time? What's the question? I
so
missed it.
this circle that
Oh,
we
the circle. Yeah, the circle is the plasmid. Yeah, one
okay,
plasmid.
is one plasma cell,
No,
or
not a cell, not a cell. A plasmid is circular DNA that's found in the bacteria, right? So
okay.
yeah. So but in this tube there are like million copies of these plasmid. Okay, it's not just but like on a molecular level, each plasmid you will put a gene in it.
So we do this so many times in that like
In the tube,
solution. Yes.
in the solution. Yeah. Well, you don't have to do many times. You just mix it together, right? So like
Yeah, but it happens automatically too many times, right?
that's right. So as soon as you mix the cut genes with the plasmid, with the cut plasmid, they both have to be cut first, right? You understand that? And they have to be cut
Yes.
with the same enzyme, right? If you cut with a different enzyme, the sticky ends are not compatible, right? Do you know what I mean by that?
Yes. Yes.
Okay, yeah. So like, okay, for those of you who are not sure, right? For example, don't copy this down. But let's say I have another gene here. Okay, but then this time the ends are like CCC, GGG, right? GGG, CCC, like that. Okay, don't copy this down, just watch. Like if you cut this thing, then the CG is not going to be binding to the AT, right? It's not complementary. Okay, that's why the flanking sequence in the gene must be the same recognition site as the one in the plasmid. Otherwise, you won't be able to connect them together. You know what I mean? Okay, so as long as you cut the plasmid and the gene with the same restriction enzyme and you mix them together, they will find each other. The gene is going to go into the plasmid, and then you just add the glue, the ligase, and you will be able to close it. And now the gene is in the plasmid. Is that okay, or it's very confusing? Anybody have questions? All right. So then we're gonna feed this to the bacteria. We'll feed it to the bacteria. Feed to bacteria. And so now the bacteria is gonna have the plasmid in it. And whatever the gene is, let's say the gene is insulin, then now the bacteria is gonna pump out insulin. Okay. And then you can collect it from the solution. That's how you do it. Okay. But the challenge is, how do you know if the bacteria have accepted the plasmid? Because you cannot see this with the naked eye, right? Like in the solution, everything is like clear, right? Like how do you know if it had accepted the plasmid? So that's what we're gonna talk about next. But before I move on, I just want to triple check that we are okay with this concept. Why we're doing it, how we're doing it, and what are the ping letters. It's okay if you have a question. Someone else probably have the same question, so just ask. Okay. All right. So let's go back to our PowerPoint slide. That was inserting a gene into the plasmid. Now let's take a look at a plasmid. Right? This is a very simple plasmid. The ones that you buy online, they are actually really complicated. Let me maybe show you like an example of this. Yeah, like something like that. Okay. Hold on. Let's just loading. Yeah, so like that, that's like if you buy it from, what is this? How do I know things? Okay. If you buy it from like a biotech company, it comes like this and it has many things in it. Okay. Now I'm gonna come back to this in a second, but let's take a look at like a basic one. Okay, so I strip it down to just like basic things. So a plasmid is gonna have an ORI, that is basically the origin of replication. Origin of replication. In other words, transcription is going to start at that point. And you have to have origin of replication all the time. And then you're going to have a bunch of things over here. Okay. Let me choose a different color. You have this PST1, ECOR-R1, BAMH1, and alpha-1. All these blue stuff, okay, they are restriction enzyme site. Now, it's important to have more than one insertion site for your gene. Let's go back to the notes for a second. Over here, our gene has the AAATTT on the side, but we actually don't control that, right? Like whatever the flanking sequence are, like it depends on what it is in the body. So we cannot guarantee that it's going to have AAATTT. So if it doesn't, then we have to find another site that's going to match the plasmid. If your plasmid only have one insertion site, then it's very limited in terms of what you can insert. But by having multiple insertion site, like, okay, let's say you don't have the AAATTT, maybe you have the recognition site for PST1. So I treat the gene with the PST1, and I treat my plasmid with PST1, it will cut right here, and then the gene is going to go in this position. If it has the same restriction enzyme site, recognition site as equal R1, then we can insert the gene here, or we can insert it here, or we can insert it there, right? So by having multiple restriction enzyme site, it gives you more option in terms of where you want to insert your gene. Sometimes another thing to consider is this. In this gene, even though you might have the flanking sequence that matches the sequence in the plasmid, you might not be able to use it if the same sequence show up within your gene. If you have another AAATTT inside your gene, that's no good, because when you do the restriction enzyme treatment, yes, you're going to create the sticky ends in the flanking sequence, but you're also going to cut your gene in half, right? We don't want to destroy the gene. All right? So that's why you need to have multiple insertion sites. And if we go back to this complicated map, all right, you see there is the origin of replication, right? And then all these black ones, those are the restriction enzyme site. Yeah, you see there's like, do we recognize any of these things? Maybe, maybe not. But there, right? There is eCore N1. It's similar to eCore R1. It's also from E. coli, but it has a different recognition site, right? But all these black ones, those are the restriction enzyme site. Okay. Now, what about? Hold on, my mouse is being a little bit wonky here. There we go. Okay, what about these things here? What about just looking for the right color here. What about that and that? Well, these are called selection markers. Selection marker. Sometimes the selection marker is gonna make the bacteria produce a particular color if they accept the plasmid, and then you can visually see it. Remember that picture I showed you where there's like blue and white? The blue ones are the ones that have accepted the plasmid. Okay, so those are the ones you want to scoop up, and then you want to mass produce them. You can just scoop them up and put them in a nutrient broth, and then they will multiply themselves. The white ones are the ones you want to ignore. In some other cases, you can select them by having an antibiotic resistant gene. So over here, AMPR is ampicillin resistant gene. And then this one is tetracycline resistant gene. In other words, if the bacteria accepts the plasmid, they will be able to gain antibiotic resistance for either ampicillin or for tetracycline. So let's take a closer look at that. I'll give you another note, but write down what you need first, and then we will talk about it. All right. So let's simplify again. Let's say we have a plasmid. Okay, plasmid right here, and I'm gonna put the AMPR here, AMPR, ampicillin resistant gene, and then I'm gonna put like the gene of interest here. This is the gene we're trying to amplify, that we're trying to make the protein from. Okay, this is the gene of interest. Okay. Gene of interest. Okay, the whole point is to feed this to the bacteria so that the bacteria is going to produce the insulin, right? For us, or whatever it is that you're trying to produce. Okay. So you're going to basically have a tube of bacteria. You're gonna give the plasmid to them. And there are a couple ways to encourage them to take it up. You can heat them up a little bit. You can shock them with a little bit of electricity. That doesn't matter, okay? But the point is that you can encourage them to take it. But not all of the bacteria are gonna take it up. Okay? There are only some that will take it up. And some would just like ignore the plasmid and carry on, right? So how do you choose which one has accepted the plasmid? Well, what we're gonna do is this: we're gonna have a plate. Okay, this is a agar plate. Agar plate. Let me Google it and show you what it looks like. Agar plate. There we go. These are agar plate, and you use them to grow bacteria. Okay, it's like full of nutrients. And they are not cheap, and basically with all the basic nutrients to support bacterial growth. Okay, so you basically add the solution to the agar plate. So this is just basic nutrient. Okay. So do you expect the bacteria to grow or not grow? Do you expect them to grow or not grow?
Yes.
Yeah, they will grow, right? They will grow. So all of it will grow. Right? All of it will grow. Okay, regardless of they have taken up the plasmid or not, they're gonna grow. Okay, so it's gonna grow so much we call them lawn growth. Right, so basically the bacteria is gonna be like all over the place. They're like, yeah, let's grow. Okay, free food. All right, so they will form all these colonies. Okay, so this is just a control for your experiment. It feels like so long ago, right? When we first met, we talked about control. Control is have a reference, have a baseline to compare to, right? So that's what it looks like if the bacteria is allowed to grow. Right? And then you're gonna have another agar plate, another plate. This is in the agar plate. But this time there is basic nutrients plus basic nutrients plus ampicillin. Basic nutrients pump ampicillin. So what happens if you plate the bacteria? You expect lots of growth, no growth, some growth. What's the situation?
Ampicillin will kill the bacteria, right?
Ampicillin, yeah, it's an antibiotic. It kills bacteria. Okay. So do you think no growth, some growth, lots of growth? Excuse me. Anybody?
I
Well,
think Professor not grow because of
yeah.
ampicillin.
Okay, yeah. So the ampicillin is gonna kill off the bacteria, right? However, if the bacteria has taken up the plasmid, then they will have gained ampicillin resistance. Okay, so some of it will grow, not a lot though. Okay, only bacteria that has taken up the plasmid will survive because the plasmid gives them ampicillin. resistance. Right? If they haven't taken up the plasmid, they will die. And that's how we know. Right? The only thing that's growing here are the ones that have accepted the plasmid. If they accept the plasmid, they have resistance to ampicillin, allowing them to grow. But more importantly, they have the gene that we want them to translate. Okay, these are the ones that will help us produce, for example, insulin. Okay? Any questions? Are you sure? Ask anything. There must be some, you know, some confusion somewhere maybe. Okay, I'll take your word for it. So let's take a look at this question. A gene is inserted into the plasmid using E-core-R1. The plasmid is transformed into a tube containing E. coli. That means you feed it to them, okay? Transform, that's what transform means. You culture all the E. coli on a plate containing tetracycline. What results do you expect? Right, so let's take a look. This is the plasmid. We're gonna insert the gene using E-core-R1. So looking at the plasmid map, you can see the gene is gonna go right here, right? Gene's gonna go right there. And if you do that, and you feed it to the bacteria, and then now you grow them in the presence of tetracycline, this plasmid has a tetracycline resistant gene. So what do you think will happen? Do you think they will grow, die, some grow? Let me do the poll. Okay, I only have 2 answers. Come on, you can do it. Choose something. Is it going to grow or not grow? Okay. Well, it will grow, but only small portion will grow. Okay. Why not all of it will grow? Well, because not all of them are going to take up the plasmid, right? Even if you feed it to them, it doesn't mean they will all take it. Okay. Only a percentage of the bacteria are going to take it up, and only those that has taken it up are going to grow. Okay? So small portion will grow. Okay, how about now? It's the exact same question, but now instead of inserting using equal R1, we are inserting using small one. Okay. Do you think it's gonna grow this time? Okay, well, not much response. Let me explain. So if you insert using SMA1, small one, the gene is going to go right here, right? Okay. In fact, the gene is going to go right in the middle of the tetracycline resistant gene. Right? So inserting a gene using SMA1 will interrupt the tetracycline resistant gene. And when that happens, the tetracycline resistant gene is no longer working. So even if the bacteria has accepted the plasmid, it will not protect them from the tetracycline. Because again, the resistant gene has been interrupted. It's not going to protect them. So all of it will die. Nothing will grow. Okay. Anything you want to ask before I move on to the next concept?
So, Vincent, you mean the SMA1 will cut the molecule of tetracycline?
That's what it will cut right there at the tetracycline resistant gene, and then you will insert your gene right in the middle of it, right? So now the tetracycline is gone.
Okay, thank you.
Yeah. Okay, so there are more questions in the study guide. Make sure you try them out. Okay, moving on. The next biotechnology that we're going to look at is DNA fingerprinting. Right, DNA fingerprinting. So let's go through some of these things. A polymorphism. A polymorphism is any difference in a DNA sequence that can be detected between individuals. Okay, so again, like we are 99.9% identical with any other humans. The difference between any of us, the 0.1% difference, that's basically a polymorphism. And it can occur in both the coding as well as the non-coding regions. It could occur in the intron or exon. It could be anywhere really. Sometimes in the non-coding regions, variation might exist in something called STR, short tandem repeats. So short means like couple nucleotides long. Tandem, it's one after the other. So these are short nucleotides that are repeated one after the other. So for example, here we have CAG, 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 CAG. So it's CAG repeated 4 times. That's how we write it, okay? So sometimes these repeats, I mean, most of the time they don't cause any problem, but sometimes they can cause problems. There is a disease called Huntington disease, which you might have heard of. It's a neurodegenerative disorder, you know, like Parkinson, Alzheimer's, they're all neurodegenerative disorder. And there's a gene called HTT. And in the HTT gene, there is a tandem repeats of CAG. Normally, the CAG is repeated 10 to 35 times within the gene. However, people with Huntington disease, the CAG can be repeated 36 to 120 times. Okay, so what happens is when these things repeats itself, CAG, 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 right, and your polymerase is trying to copy it, it makes a mistake. It might slip during the process, and therefore accidentally put in like an extra one, or it might miss one. Right? So the length of the tandem repeats might vary from one generation to the next generation. So what happens is, let's say the person has like 30 repeats, right? And then when they make their gametes, that increase to like 34, And then the next generation, maybe there's another mistake, and then it pushes it over the threshold of 35, then the person might develop Huntington disease. Okay, this is actually a dominant condition, dominant. So if a person has Huntington disease, their kid might have a 50% to 100% chance of getting it. Okay, depending on whether the person is homozygous or heterozygous. And the symptoms of Huntington usually don't show up until after 40. By then, people would have already had kids, right? And so that creates like a little difficult situation. Right? Do the people want to go get tested? This is something you can actually test for, genetic testing, right? So do you want to test and find out that you are okay or not okay? And if it's not okay, like you can make plans ahead, but it's probably gonna ruin, your everyday life, right? It's gonna change it dramatically, right? And so there's some ethical dilemma right there. Okay, so that's what happens. But not all tandem repeats result in disease. And we can actually use the difference in length for these repeats to do what's called DNA fingerprinting. So for example, person 1 might have AGAT repeated 12 times, right, on chromosome 6, for example. You would find the same tandem repeats on chromosome 6 at the same place for person number 2, but it might be shorter. So instead of 12 repeats, it only have 5 repeats. And then you can look at another tandem repeats, right? This one repeats 15 times. 15 times is the same. But when I look at the third one, it's 30 and 20. Right? So if we compare all the STRs in a person, then we get a very unique combinations for all these STRs. And that's why that's called DNA fingerprinting. Now, to understand how to do STR analysis, we have to talk about what's called, electrophoresis. So electrophoresis, gel electrophoresis, that is a technique used for separates DNA based on its length, okay, or size. So you can separate long DNA from short DNA. That's what the technique is used for. It's called gel electrophoresis. So there are a couple of things here. There needs to be a power supply, and you're gonna hook it up so that the negative is here and the positive here. And over here, that's gel. I'll show you the gel in a bit. But this is the top of the gel, these slots. That's where you're gonna load the DNA. Low DNA sample. I'll show you how that's done in a bit, okay, in a video. That's where you load the DNA sample. Now, DNA backbone is negatively charged because of the phosphate, right? The PO4 -3. So when you apply a current from negative to positive, it will push the DNA down the gel. So the DNA is going to move through the gel like that. Now you're going to need a buffer solution to help conduct the electricity, conduct the current. Okay? So over time, the DNA will slowly move down the gel. Now let me show you how the gel is made. Okay, so here's the gel. It's like Jello, basically. Right, let me see if I can skip through it. So this is the cast. Okay, you are going to tape it off. Okay, I'm just gonna skip to the interesting part here. So now you have a nice well, and then you pour in the gel solution. Okay, the thing, this is called agarose gel. Okay, it comes in a powder form, and then you add some buffer solutions to it, and then you microwave it. Okay, for like 1 minute, so it helps dissolve the powder, and then you pour it into the cast here, and then there's the comb. Okay? The comb is gonna make a little indentation in the gel. It creates like a little well for you to put the DNA in later. Okay? And then you let it sit there for like half an hour. You pull out the comb, and you can see those are the wells. Right? That's where you're gonna load the DNA. All right. So actually, it's quite fun. But unfortunately, we don't have the budget to do that in class. So I just have to show you a video. So the agarose, this is a gel. And if you zoom in the gel, the microscopic structure looks like this. It's like a mishmash. Okay. So when the DNA try to go through these mishmash, if it's a really long DNA, it gets tangled up and it's not able to move very far. But if DNA is really short, it's able to go through quite quickly. So one time I took my kids to this playground, and they have this thing in the playground. I don't know if you guys seen this before, okay? So you're supposed to get in from the top, and I guess you weave your way through these networks and try to get to the bottom, right? And so my daughter wanted to challenge me and say, let's see who can get to the bottom the quickest. Right, so I was like, oh yeah, sure, okay, challenge accepted. So we did it. Since they were much smaller, they actually are able to go through quite quickly. Right? For me, I can still get through, but I get stuck a couple of times, like literally got stuck, right? And if we only given like 2 minutes, for example, then the smaller person is going to be able to make it further compared to a bigger person. And that's what happens. You load the DNA sample here, and you let the current push it for a limited amount of time. The DNA that are shorter, it's going to be able to go further. But the longer DNA, it's going to be closer to the well. So that's going to be like a longer DNA, and that's going to be a shorter DNA. And that's how you separate the DNA based on length. Okay, let's say you have a whole tube, a bunch of DNA sample in there, some long, some short. When you lower it under the gel, this will spread them out so that the shorter ones are going to be at the bottom and the longer ones are going to be at the top. Anybody want to ask me anything? Okay. So then, you actually cannot see the DNA. Right? Let me show you another video here. This is all the equipment. That's the power supply right there. Can you see my mouse? Oh yeah, there's the power supply. You're gonna hook it up to this chamber. These are the pipette. These are the Eppendorf tubes with the DNA in it. And there's the gel. They're gonna put the gel here, right? So I'm just gonna skip to the part. There you go. That's the apparatus. The arrow tells you which way the current is going. So black goes to black and red goes to red. You can't mess that up. That's the negative. That's the positive, right? So they're explaining. So you filled it up with the buffer. The purpose of the buffer is to conduct the current. Yeah, it contains like sodium ion and stuff like that. These are the DNA sample, and so you actually have to add a blue dye to the DNA. Okay, because the blue dye is heavy, it will help the sample sink into the well. If you don't add the dye, the DNA sample is just going to come out of the well and then it just goes all over and you will lose the sample. Okay, let's take a look at how the person loads the DNA sample. Okay, so the person took all of it. Okay, and then this is the well. You put the pipette in, that's just a solution, and then you squeeze, and the dye makes it sink into the well. You see that? So that's one DNA sample here, that's a second DNA sample, and then you basically keep on loading. As many sample as you have. Gonna load a third sample. If you don't have the blue dye, then again, all of it is just gonna swoosh out of the gel, and it's gonna be lost in the buffer solution. Okay. So then you close the lid, you plug it in, you set the voltage, and then you set the time. Okay, usually, you know, it's gonna take not too long, like half an hour. And the bubble actually shows that the electric current is going. Okay, so you don't want to be, you know, sticking your finger in there. And over time, you can see the DNA is gonna slowly move down. Okay, you can slowly move down. Now we actually cannot see the DNA. Okay, we only see the dye. That it's called the dye front, front. The dye front gives you an approximate idea of where the DNA is. So the dye is right here, and the dye is really small. So the dye is here. Your DNA is roughly over here. The really long ones are probably over here, closer to the well. And if it's really short, it's gonna be somewhere here. It's not much shorter than the dye. Right? So you don't want to run the gel for too long, because if you keep on running it, if you keep on letting the current go, eventually the dye is going to come out from the other end, which means your sample is lost, right? The DNA eventually is going to make it all the way, even the long ones. If you run it for very long, they're going to go all the way to the other end, and then it's going to exit, and you're going to lose the sample into the solution, right? So you want to stop this current when the dye front is about like halfway. Okay? So that's basically it. Any any questions? I know I keep on asking you every 2 minutes, but do you understand like the process? Okay. Now you cannot see the DNA. You can see the diaphragm, you can see the blue line, but I cannot see the DNA with my naked eye. And to see it, you have to add something called ethidium bromide. So ethidium bromide is a molecule that will bind to the DNA and make it fluoresce when you shine UV light at it. Okay, it binds to the backbone of the DNA. Because it binds to the backbone of DNA, it's a carcinogen. Okay? You don't want to be getting it on your hands and then, you know, it's get into your cells and change your DNA. Okay, so when you handle this, you actually have to wear gloves. And then you stain the gel with ethidium bromide, and then you shine UV light at it. There's a special machine for that, like a chamber that you stick the gel in. And then now you can see all these bands, that's where the DNA are located. Okay, so the really short one is over there, the really long one is up here. Okay, and then you can take a picture, and usually the picture looks like this. So the first lane here and the last lane here, you can see there are many bands. These are actually called leather. You can buy them. These are pre-made DNA with certain length, certain size. Okay, so that's for you to measure your DNA sample. Like if you see a band there, like how do you know how long it is? Well, you can compare to the leather. The leather says this over at this position is about 268. So you can say this line is also 268. Right? And so the ladder gives you like a measuring tool, essentially. All right, so let's go back to the DNA forensics. Like, how do we use this DNA fingerprinting along with the STR to figure out, you know, who's the like the what I'm trying to say, who's the suspect, right? And who's the actual perpetrator. Okay, first of all, let's take a look at this first. The plasmid is treated with E-core-R1 and BAMH. When the digested plasmid is separated using gel electrophoresis, how many bands will you see? Let's think about this. You're going to take this plasmid, you're going to treat it with E-core-R1 and BAMH. How many are you going to see? How many bands are you going to see? You can try this with an elastic band if you have a hard time visualizing it. Okay. So E-core-R1 is going to cut it right here, and BAMH is going to cut it right here. So if you take an elastic band, which is like a plasmid, you cut it 2 times, how many fragments are you going to have? two fragments, right? There's a short one right here, and then there's a long one right here. Okay, so if you run the sample on a gel, you will get a band at the top corresponding to the longer fragment, and then you will get a band at the bottom corresponding to the shorter fragment. Okay. All right, let's take a quick note on STR analysis. There we go. STR analysis. So let's say we have two people here. Two people. Let's call this Bob. And I'm gonna look at, let's say, chromosome 4. Okay, on chromosome 4, there is an STR here. And this is like CCG repeated 10 times. Okay, CCG repeated 10 times. And then I'm gonna look at another chromosome, maybe chromosome 17. And there is another thing here, let's say is AT repeated 5 times. Okay, and then just one more. Let's say we look at chromosome 22, and then there's another tandem repeats here. This one is CAA repeated. Hold on, I gotta make sure the math works out. 5 times. Okay. Now we're gonna have another person here. This is Amy. And we're gonna look at the same chromosome. Chromosome 4, she will also have CCG, but the number of repeats might be different. Let's say she has repeats of like 15 times for CCG. And then for AT, it's also 5 times just by chance. And then 22, there's CAA repeated. Let me think about this. CAA repeated.
So Vincent, I have a question. Can I?
Yeah, sure. Yeah, go ahead. What's the question?
These sequences are non-codon, not right? They are not making
Yeah,
protein.
they could be in the coding or they could be in the non-coding. Yeah.
Okay.
Yeah, both is okay. All right, so let's say just repeat it 2 times. Now, try to follow the next part. Because we know where these tandem repeats are located, you can look at them in the genome browser, right? We've mapped out the whole human genome, so we know where these tandem repeats occur. So for example, for chromosome 4, what we want to do is we want to design primers that would flank the CCG. And we're going to use PCR to amplify it. Okay, so we'll use the same primer for Amy's DNA sample. We need separate primer for the AT. And then we also need like a third set of primer for the CAA. Like that. And then we're going to go PCR. So we only amplify those repeats. Gonna amplify the repeats. You're gonna get like, millions of copies of this CCG. So let's draw the gel. There's the gel. Okay, so this is a well, this is where you load the sample, right? So then I load the sample of Bob here. Now let's do some math. CCG repeated 10 times. How many base pair is that? How many base pair is that? CCG repeated 10 times. It's 3 times 10, right? Isn't it? CCG, CCG, if you just keep on counting, it's 3 times 10. So that's 30 base pair. How about over here? AT repeated 5 times. How many base pair?
15.
But it's 2 times 10. That's right. 2 times 5. That's 10. Because you're only repeating ATs, right? And how about this one? CAA repeated 5 times.
15.
15 base pair. That's right. So in your tube, in your test tube, after you do the PCR, you would have like the red DNA. They're not really red, okay, but they're the red ones that are copied from there. They're the blue ones, and then they're the green ones, okay? Again, they're not color. I'm coloring them to show you for simplicity. And then you load them onto the gel. You will load Amy sample over here at the same time. And then, yeah, go ahead. There's a question. Yeah.
Professor, I didn't understand why CAA. So repeat the 5 times. It
Yeah,
will
so
be
CAA, that's 3 base pair, right?
yes.
And then CAA, 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 and one more time, CAA. So
Oh,
3, 6,
count.
9, right? You understand now?
Okay, okay, Professor.
Yeah, no, that's okay. It's very confusing, I know. I'm so sorry. I don't know why this is so hard. So you're gonna load that thing, and look, we have 30, we have 10, we have 15, right? Which one is gonna go the furthest? Which one's gonna be able to travel the furthest in the lane, in the gel?
Blue one?
Which one? I'm sorry. Is it the red, blue,
Blue
or
one.
green?
Blue.
Blue. That's right. Because it's only 10 base pair, right? It's really short, so it's gonna be able to go really far. Okay, really far. Okay, like there. And then we have the green one, which is 15. I mean, 15 is a little bit shorter than the 10, a little bit longer than 10. So it's probably very close by. Okay. And then for the last one here, 30 base pair. That's really high up, like that. Okay. So Bob is gonna get 3 bends. Okay, let's take a look at Amy, huh? We're gonna do PCR, and then we're gonna load Amy's DNA here. Let's figure this out. CCG repeated 15 times. How many base pair is that? You can type it in the chat box, okay? Just show me you understand. So I know you can do this on the test. How many, Mary? You were saying? 4. Yeah?
45.
45. Yes, 45 base pair. 5 times 2 is 10. And then 2 times 3 is 6, right? So which one is gonna go the furthest? Which one is gonna go the furthest? Is it the red, blue, or green? Which one will travel the furthest?
Green?
That's right, because it's the shortest, right? In fact, if you take a look at the shortest one here, it's even shorter than what Bob has. So it's gonna be a little bit further down. Okay. The blue one is repeated 10 times, just like Bob's. So it's gonna be exactly same as where Bob's line is. And then we have the 45. 45 is even longer than 30. So it's gonna be a little bit higher than Bob, right? Look, this is the 45, right? This is the 30. Okay, look at the numbers. This is the 6, and this is the 15. And this is 10 and 10. So you see, with 3 different bands, 3 different STR location, you can create 3 bands. And then the bands are unique, right? But when we actually do this, we don't just do 3 STR, we do as many as possible. So you're gonna get like many, many, many bands like this over here, right? You're gonna get many, many bands. So this is the DNA recovered from a crime scene. This is DNA sample from suspect 1, 2, 3. And then you look at the STR banding. Okay. Which one is the perpetrator? Which one should we charge?
Second one, middle one.
Yeah, second one, right? The DNA looks exactly the same. You see? But you understand, right? These are tandem repeats. Okay. So that's called DNA fingerprinting. Understand? Very cool.
So Vincent, this is unique for each person?
That's right. That like it's very accurate. Your STR repeats are gonna be different from another person. It's like all of us has a barcode, right? Has a barcode, right? People who are related, they have similar STR. So we can also use DNA fingerprinting to determine paternity and maternity. Well, yeah, usually paternity, right? To determine if like, you know, who the father is, if there is a, you know, dispute, right? Okay, let's take a look at this. I'm gonna launch the poll. Please participate if you can. Okay, not bad. Right. CAG is repeated 30 times, is repeated 10 times, so it's 30 in total. This one is 45 in total, length, right? So it's asking you which one will go down the jail further. It's gonna be A, because A is shorter. The shorter it is, the further it's gonna be able to travel down the jail. Okay. A DNA ladder is loaded in lane 1, and a DNA sample treated with restriction enzyme is loaded in lane 2. If the DNA sample only has 2 recognition sites for the restriction enzyme, what is the original length of the DNA sample? Okay, so this is the plasmid. It has two recognition sites. Oh my goodness, 2 recognition sites. Oh, sorry, not a plasmid, a DNA sample. Okay, so DNA sample is like linear, right? If they don't say plasmid, it's linear. So you cut it twice, and that's why you have 3 fragments, right? Okay. 1 fragment, 2 fragment, 3 fragments, and then you load all this into the lane here, and then you want to figure out the original length. Well, it looks like we have a really short one here. That's like between 2 and 3. I'm gonna say it's 2 here. Okay. Like in reality, it's not gonna be exact, okay? And then this is gonna be 6. And what do you think that one is? What's a reasonable number for that one? How long is that piece?
9.
Yeah, 9. That's a reasonable number. So if you add them together, 9 plus 6 is 15, plus 2 is 17. So the original length is 17 kilo base pair, right? kilobase pair, kb. I guess, you know, you can actually go like 2.5 here to be more accurate, I suppose, halfway between, right? So what is that? That's like 17.5 kb, kilobase, right? Kilo means 1,000, right? So that's what it is.
Vincent, why some of the bars are like thinner than others? Like
Why are some
does
why
it?
are some bands thinner than the others?
Yes.
Yeah. That's a good question. Okay, don't worry about it, but then I can tell you. Yeah. Sometimes it's because of this. Sometimes it's because of this. Let's say you have the DNA sample, and let's say it's like this, okay? Let's say you cut it here, and then you cut it here. Okay. So how many fragments do I have?
3.
3, exactly. So this one is really short, right? But the other 2 are same length. So when you lower it on the gel, you're gonna get like a thicker one because you have twice the amount of DNA, and then you have a thin one there, right? Okay, so that's why
Oh,
sometimes some are thicker than the others, but don't worry about that, okay? For what we're doing, it's already confusing enough, but very good observation skill there. Okay, so that's that. Oh, by the way, have you guys received an invitation to do like the course evaluation yet? Do you guys know what I'm talking about? Like a survey?
Yes, Professor.
Yes, okay. So please take a moment and do that. Okay. For all the courses, they are anonymous. We don't know who's saying what. Okay, so you can confidently say what you want to say. And those are really important for us because we use those information to do like an annual review process. Okay? I'm the one who have to write the report, and what's really helpful when I write a report is to gather all the comments from all the courses, and then you know, make recommendations of what we can do better, or you know, maybe there are some things we are already doing well and we want to know about it. So your opinion is very important, especially when it relates to like the online learning environment, right? So I know there are still things that we could improve on, but we don't know unless you tell us. So please take a moment and do those survey, not just for my course, right? For all the courses. Thank you. All right, we are almost there. We will do what's called RFLP. RFLP analysis. So this is called restriction fragment length polymorphism. So on the left side, we have a crime scene DNA. Okay. And we're basically going to take the DNA and we're going to treat it with restriction enzymes. So the restriction enzyme will cut it. Let's say the restriction enzyme recognizes the CCGG. So in this DNA sample, there are 2 such locations, and it will cut it twice. And that's going to create 3 bands, 1, 2, 3. But remember, there are a lot of silent mutation throughout this body, right? So if someone else, the suspect, he has a mutation here in one of the restriction sites that causes the palindromic sequence to disappear. When you treat this DNA with the same restriction enzyme, you will only cut it once, and there's only 2 bands. Okay? So by treating a DNA sample with restriction enzyme, we can see whether the fragment lengths are the same or not. Okay? And if they're the same, then we can say it's the same sample. This is an oversimplification, right? There are like many restriction sites throughout the entire genome, right? So for 2 different people, it's very unlikely, right? It's actually not virtually impossible for them to have the exact same site. Certainly not for multiple restriction enzymes. Okay, if you do it for one, maybe it's very close call. But if you expose it to multiple different enzymes, right, you're not going to get the same thing. So I think I might have like another slide here earlier, right? Like this is basically what happens, right? You get all these fragments, and then you can compare and see which one are the same and which is not, right? Okay, that's all. We're just gonna go through the last part of the lecture. The hardest stuff is over. Right now, it's just very basic things. Right, so last part is called gene therapy. So the basic concept of gene therapy involves introducing genetic material into a person's cell, and that genetic material is going to help fix some kind of problem. Okay, it's going to help alter the outcome of a disease or completely cure a disease. Right, I'm going to show you the examples later. But depending on which cell, what type of cells you are modifying, the outcome can be different. Okay, we have what's called somatic gene therapy. So somatic are bodily cells, except for the germ cells, right? So if you modify the somatic cells, those modification will only affect the person and not the future generation. The modifications will not get passed down to future generations. So on the other hand, if you change the germline cells, germline gene therapy, those will become the egg and the sperm, and it will affect future generations. So ethically speaking, germline therapy is currently not approved, right? Because we don't want to be changing future genetic information, right? First of all, the future generations have no consent, like your kids doesn't even know about this modification. And secondly, we don't know like if there's any kind of long-term negative impact, right? So somatic gene therapy is the only one that is approved. Okay. But like, you know, I don't know. I'm sure there are some places where this is being violated. Okay, there is someone maybe secretly doing germline gene therapy. We don't know. So what's the point? The point is either to replace a non-functional gene, suppress an overly active gene, or modify the clinical course of a condition. So let me go through each one of these in more detail. The first one is very straightforward. Okay. We try to replace a defective gene. So you have a non-functioning gene, we have a functional copy, right? And we know we can clone it from like a healthy person, right? For example, and then we would deliver it to the gene to the patient cell, and then once the functioning gene goes in, you will fix it. So something like sickle cell anemia, right? We know it's caused by a mutation in the hemoglobin gene. You deliver the correct gene, they will make the correct hemoglobin, then you fix the disease. Okay. So these are typically ideal for loss of function in the protein and has to be recessive. Why is that the case? Well, if the disease is dominant, even if you put in the functional gene is not going to cover up the dominant disease, right? Okay. Dominant is always going to be expressing itself. Okay. In recessive, let's say it's little s, as soon as you introduce the big S, then you fix the problem, right? So these have to be recessive diseases. And it is only successful if the effects of the disease are reversible or have not resulted in lasting damage to the body. Okay. There are some diseases that causes neurological damage during development. Okay, so if the person is born with this disease and during the critical years of brain development, they have the symptoms, right? So if you try to use gene therapy to fix this by age 10, all the damage that has been done from 0 to 10, it's already there. You cannot reverse it. You can only prevent further damage. So those diseases are not really ideal, right, for using gene therapy. We want things that are reversible. So sickle cell anemia, beta thalassemia, that's a nonsense mutation in hemoglobin. You're missing a whole chunk. Right? Hemophilia is bleeding disease, right? There are many factors, proteins that helps you stop the bleeding in the body. If some of them are mutated, not working, you need to fix them. And by introducing the correct version, the functional gene, you will be able to do. So I think there are a few successful cases of gene therapy already. I've heard about people who are blind and now like born blind because of a missing protein in the eye receptor. And they are able to restore vision. There are people who are born deaf because of there are some cells in the ear called hair cells, and they are responsible for picking up sound. You will learn that next semester. But if they don't have the protein to make those receptors, then they will be born deaf. And I've heard about like a podcast that talks about using gene therapy to fix that, and they're able to hear for the first time. Okay, second option is to suppress a gene. Sometimes a mutation can cause the gene to become overly active. We talked about this before. It's a gain of function mutation, the green lights that are always on, right? Like an oncogene. So we can deliver a gene that will go into your cell, and we call this like a blocking gene that basically will shut off the green light. Okay, it will turn off those overly active genes. And by turning off these oncogenes, maybe you can suppress the cancer, right? Lastly, we can modify the clinical course of a condition. So in this case, you can do it in two ways. Number one, you can insert DNA, you can insert genes that will trigger the cancer cell to die. Right? We call this like a suicide gene. It will trigger cell death in the cancer, and the cancer cell will basically kill itself. You can also insert a gene that causes your cells to put special markers on them, cause the cancer cell to put special markers on the cell membrane, and your immune system will recognize these surface markers, and it will attack them for you. One of the problems is that the cancer cells arise from your own cells. So your immune system have a very hard time distinguishing what belongs to the body and what belongs to the cancer. Right? The cancer is kind of like camouflage, hiding right behind your own cells. And by inserting these markers on them, it basically makes them glow. And the immune system, the immune cells, the white cells will be able to find them and kill them. So here is the suicide gene, deliver it into the disease cell, cause it to die. Or you put a marker gene and now you have all these glowing beacons on it, and the white cells will come and eat them.
I have a question, please.
Yeah?
Are these treatments are being done in
Well, these are still in the experimental phase, but as many of these are actually in clinical trial, and these has been done. Okay, you can actually in some cases take the white cells out of the body. You can educate them in the petri dish by again, you will learn this next semester, but you can educate them by showing them what these glowing dots are like, right? And then you put them back into the body, and they would kill the cancer. Okay, but it's still early stages. There are a lot of problems with these things, and I'll tell you some of the problems in a bit.
So chemotherapy, some of these treatment, chemotherapy.
Okay, chemotherapy means treating the cancer with drugs, right? With medicine. This kind of stuff, it's called immunotherapy, because you're using your immune system to help it, right? And then you can supplement the immunotherapy with the gene therapy, right, to make it even more potent. And sometimes you genetically modify the white cells before introducing it back into the body. Right? So that's actually my next slide. Right? Ex vivo means outside of the body. So you extract the cells from the body, you make some modifications to them, right? Teach them how to recognize the cancer cells. Okay, teach them how to fight off the cancers, and then you re-implant those cells into the body. All right, so this is like you have more control over it because everything happens in the petri dish, and if something goes wrong, you can always like remove the cells. You have more control, right? But in vivo, we are trying to use a vector, usually like a virus, to help deliver the DNA to the cells. Now that is a lot harder to do Okay, like how do you know the DNA is gonna go to where it's supposed to go? Which brings us to the next slide, right? A lot of the challenges. The biggest challenge of gene therapy is targeted delivery. Let's say we talk about the suicide gene, right? You want it to go to the cancer cells only. But how do you make sure of that? Like, what if the suicide gene accidentally went into your healthy cells? Then your healthy cells start dying. Okay. Same thing with the markers, right? Like, what if you deliver the marker and it's supposed to go into the cancer cells, but then it shows up on your healthy cells? That's actually what happened in one of the articles I read. They were trying to do immunotherapy and gene therapy for someone with skin cancer. And what ended up happening is, yes, the cancer regresses, but at the same time, the liver is failing because of the off-target effect. The immune system attacks the cancer cell, but at the same time, they also attack the liver cell. Okay, maybe there's some similarity between the skin cancer and the liver. Okay, so that's off-target effect. So sure, we keep the cancer at bay, but the person is also dying because the liver is failing. But as soon as you take the person off the therapy, the liver function returns, but so does the cancer, right? So there's the target delivery. Immune response, right? You might have undesired off-target immune response, as I just told you. But when you're delivering with a virus, the immune system might also reject that, complicating the insertion effort. It might interact with other genes in an unknown way. There's so many genes in the body. How do you know you will only turn off the oncogene that you intend to turn off? Like, what if it accidentally turned off some other thing? Maybe it turned off a tumor suppressor gene, then we create a whole new set of problem, right? So it's at this point, it's very hard to control. Okay? You might have heard of CRISPR technology. So that is like revolutionary. It allows us to make changes in a very precise way. So I think in the next 10, 20 years, this is going to be it's going to mature a lot, and we will have a lot more success. However, the cost remains to be a very big hurdle. Okay, right now people are just doing experiments, so a lot of time these things are like sponsored, right? Because if it works, they collect a lot of data. It could mean much more revenue down the road, right? But these things are not cheap. For the blindness that I was telling you about, it causes $600,000 per eye upfront, and then every year you have to pay an additional 100 grand just to maintain, make sure everything is okay. And there's no guarantee these things are going to work. Like, what if you pay and then it ends up not working, right? So there is certainly, and it's US dollar I was talking about, right? So there is a lot of problems surrounding accessibility. Like, is this just a treatment for people who are rich, for people who can afford it, right? Like, is insurance going to pay for it, right? And again, even insurance pay for it, not everybody has insurance, right? Coverage. So there's a lot of challenges and a lot of hurdles. Yeah, you can look up successful gene therapy on Google, and you will find that many of these are actually well underway. Right? I think just last month I read about a successful sickle cell anemia treatment. Okay, so something like that, like sickle cell anemia, is going to be very common for many people, right? So the cost for that is likely to come down if it's successful. But a lot of these genetic diseases are very rare, right? And like the blindness, like the hearing, those are not very common conditions. They are ideal targets for gene therapy, but because it's not very common, even if it's successful, right, it might require like tailored solutions for each patient. Everybody might different, and that could drive the cost up substantially. And that is it for biotechnology. I know there are a lot of things you still need to sort out on your own. I will try to take some time next Monday to go through the lecture study guide with you. Okay, you go through it yourself first, but I'll be happy to take up some questions with you. Is it possible to upload lecture A study guide? I thought I did. Is it not there? I know you emailed me over the weekend. But I thought it's up already. Is it not?
Actually, you posted the class note, but I am talking about the study guide.
The study guide. Okay, let me take a look here. Okay, sorry about
Yes. No, no,
this.
it's okay.
Is 202 content study guide number 7. Oh, okay. Totally my fault. So sorry. So that's lecture A, including the tutorial. I'll do number 9 as well, right there. And let me just do number 10 as well so I don't forget, okay? So they're all there now, all right? We were supposed to have the quiz on Tuesday, is that right? If I remember correctly, let me just double check.
Not Tuesday. You don't have Tuesday class.
Oh, sorry, yeah, Monday, I mean, the end of semester, my brain is not really working anymore. Hold on, let's see. This is like that. You guys are online. The other online. Yeah, you were supposed to have it on Monday. Why don't I move it to Wednesday? Okay, so you have more time. Yeah,
So yes, Professor,
yeah,
thank you.
Monday. I will answer any question you might have for 8 and 9. Maybe we spend half an hour doing that. But you have to ask questions, okay? Because I don't know what you don't know. Okay, so bring questions, and then we'll take it up, and then I'll move the quiz to Wednesday. Okay.
Sir, I
Thank
have
you.
to
Thank
tell
you.
one thing.
Sorry, what is that? Yeah, go ahead.
Yeah, I have to tell one thing.
Mhm.
At Wednesday and Thursday, we have Eid. Eid
Oh yeah,
celebration.
right. It's
Is
end of
it
Ramadan, right?
possible to open
Yeah, sure.
the
I'll do it.
till
How about
Friday?
Wednesday to Friday? You want Wednesday to Friday? Yeah.
Yeah. Then it will
Okay,
be.
sure. So I open it on Wednesday, and then for those of you who are celebrating, that's fine. Okay, you can do it like, and this goes for other people as well. Okay, like if you want to do it later, I'll give you until Friday. Okay.
Okay, thank you.
Yeah, and then we'll do a review next week, and then the final test, and then that's it. Okay. Are you guys also taking